My name is Barney Graham, and it's my great honor today to get to introduce Dr. Jose Antonio Melero, who is one of our co-awardees for the Chanick Prize this year. And Jose and I are connected in many different ways, and I'll go through some of them, but we have both personal connections as well as professional. Jose trained in Madrid and got his PhD in biochemistry there before going to the Imperial College in London for a postdoctoral fellowship in tumor virology. He continued that work at NYU Pathology Department and then moved to the National Center for Microbiology at the Institute of Salud Carlos III in Madrid, where he has been there ever since as director and is now the head of the viral biology group. He's received the Spanish Society of Virology Prize in 2011 and received an honorary doctorate degree from Uruguay in 2014. I just want to say a couple of things about Jose because we both published our first RSV paper in the same year. I won't tell you exactly when that was, but we've been at this together for a long time and coming somewhat from different angles. Maybe me more immunology, clinical thinking, translational science, and him more as a virologist, but I was drifting more toward virology, and he was a virologist drifting more toward immunology, and we ended up meeting on the F protein, on the F glycoprotein, where he has made seminal contributions to our understanding of its antigenicity, monoclonal antibodies, its structure, since the early 1990s. For more than 20 years, he's been helping us see through how the F glycoprotein works, among many other things. I'm just going to talk about F because there's so many things besides that. And one of the things that I love about Jose is that he just has a beautiful mind. And it's the basis for his sense of humor that everyone should experience and enjoy, but also because he has done very, in some ways, simple but profound experiments and has been able to interpret how F works, how it looks, and how its antigenicity and everything about F from very fundamental work, and it's just his vision of and ability to see things in these experiments that have been so impressive to me. And he showed us the first image of F, several years ago, and now I think he's going to tell us more about F and other things and describe his career in much more detail. And I want to bring him up here so I can hand him off this prize that he's won. Give this to you. You want me to help you figure it out? Maybe. Oh, whoa. Oh, it's upside down. Yeah. Thank you very much.
Wow. Thank you very much, Barney, for this kind presentation. For many people in the audience, maybe that I look like an imposter. And I may be even an imposter. Anyway, I would start by thanking all those that voted for my nomination to this award, okay? I am obviously very honored and I must say also overwhelmed by it, okay? So I'll try to tell you some stories about RSV and me, okay? And I will start by saying a few words about the person that obviously has the name of this award and who is responsible for all of us being here together today. Dr. Robert Channock, as you know, he was the first that isolated the virus that we are concerned today, RSV, from a human being, and also demonstrated the relevance of this virus for viral infections, particularly in children. And I met him in a few occasions. The first one was in this place, which is not Bariloche. It's the equivalent to Bariloche in the French Alps in the south of France. It's a place called Annecy, and there the Melero Foundation has a place where they organize scientific meetings regularly. And in 1990, they organized this meeting where I think I met also for the first time Peter Openshaw in animal models of respiratory virus infection. And I gave a talk there, and after the talk he came, Dr. Channock came to me and said, Jose, you know, antibodies are important for this virus, so you are working with antibodies, keep going. And we did. Also, I would like to mention the people that have received this award before, starting from Brian Murphy, that received the first of these awards from Bob Channock himself. Then Caroline Brishall, Gail Wards, the two Peters, Peter Collins and Peter Openshaw. And the last one was Barney, that received this award in the meeting in South Africa, the previous meeting, the meeting previous to this one. He got the largest picture because he's also largest. Oh, sorry. Not yet. But not because the main reason for this large picture is because I wanted to show this structure that is shown in the screen behind him, and is the protein that he was referring to before, is the F protein, okay? And some people may wonder whether it's the pre-fusion, the post-fusion. It's none of those, okay? It's actually an intermediate. It's an intermediate which is called pre-hairpin, and which I will say something more about it later. And when I received the news from Fernando about the nominations and this award, I was really very glad to hear that I was going to share this award with Ruth Caron. Ruth and I had been attending these meetings for a long time already, so we have met regularly in these meetings, but also in other places. And over the years, we have become very good friends. We haven't collaborated directly yet, okay? But we have talked about death and life and everything, you know, besides science. So now you understand that I am overwhelmed, and of course I am very honored to become a member of this exclusive club, okay? So, I will start by going back to the time that Barney was referring at the very beginning, not 25, but 30 years ago, okay? In which one of the first papers we published was this one, and already we were in the title, in the title we were already indicating some something about the antigenic structure of the RSV glycoproteins, and some striking differences that we found and we didn't understand so much at the time. And these differences are indicated in this table. So In the lab, we had already isolated a panel of monoclonal antibodies recognizing either the F protein or the G protein. In our nomenclature, F means fusion protein, G means G protein. And our monoclonals, they always have the F or G. So you can imagine, you can already work out immediately whether they are specific for the F or the G. Okay. So in this case, we isolated some viruses that escape neutralization to one of these antibodies, 47F. 47F, which turned out to be similar to the more commonly known palivizumab antibody, okay? And these viruses, as you see here, they had lost reactivity with the antibody that we used for selection, and also another antibody that competed with 47F for binding, so they were classified in the same antigenic group. And that's what one would expect for a escape mutant. This is the classical behavior of escape mutants from most viruses ever. But reacted normally with all the other antibodies, okay? In contrast, these viruses that were selected for resistance to this antibody recognizing the G protein 63G, they had lost reactivity with the antibody used for the selection, but they had also lost reactivity with most of the antibodies that we have, even if they had been classified, at least by antibody binding, in the different antigenic sites. So this was a big surprise, and we didn't understand what was really going on until we sequenced these viruses. In the case of the mutants resistant to 47F antibody, what we found were single nucleotide changes that introduce an amino acid change. For example, in these two mutants had a change from asparagine to tyrosine at residue 262, and these three others, they had this other mutation. So this was what it was described for most of the escape mutants in a whole variety of viruses that were already characterized and published. In contrast, the viruses that we isolated and that were resistant to antibody 63G, as I showed in the previous slide, they had lost reactivity with most of the antibodies that we have. And these viruses, they had, instead of a nucleotide substitution, what they had was a single nucleotide deletion. It was an adenosine deletion in an oligo-A tract that was in the middle, in the body of the G gene. And this nucleotide deletion introduced a frame shift mutation that changed, as indicated here, it changed essentially the sequence of the C-terminal third of the molecule. And this was amazing because, I mean, for a protein to change 1/3 of the sequence and still being functional, it was something that it was really striking. We isolated more mutants with antibodies against the G protein. We eventually found mutants that behave more traditionally, like these ones that they had a single amino acid changes. But we also isolated mutants that had premature stop codons that, again, in this case, the mutations deleted a large part of the C-terminal part of the G protein. And again, these mutants were viable and they replicate normally in tissue culture. We even selected mutants that, like these ones, that had not just one nucleotide substitution, but multiple nucleotide substitutions, and eventually, or actually, most of these sequence changes were A to G transitions that resemble the hypermutations that were described, for example, in HIV. And so we were describing all these sort of very strange mutations that the G protein could accommodate without losing its functionality, and we were really happy until our friend Ruth Caron came along and said, well, why don't you forget about all this? I mean, you can just forget about the G protein, because she described these mutant viruses that had big deletions that eliminated essentially all the SH and G sequences in the viral genome. And these viruses could still replicate in tissue culture, although they were attenuated in vivo, which is what they were looking for, actually. But after the first shock, we thought that what geneticists say, if either use it or lose it. So if the G protein is still there, okay, it must be useful. And in fact, it turns out that these viruses that were first isolated as a spontaneous mutant, they were then reconstructed by reverse genetics, by Peter Collins, Mark Peoples, and other people. And these viruses, they do replicate in tissue culture, but they are not as good as the wild type. And as I said, in vivo, these viruses are attenuated. So all the information that we gather with the escape mutants and the monoclonal antibodies allow us to at least propose some sort of structure, very basic structure of the G protein of the virus. The G protein is a type 2 glycoprotein, so it has only one hydrophobic region near the N-terminus, okay? And in the ectodomain, it has these two regions that resemble the mucins that are produced by epithelial cells, and they are extremely variable. And the strain, so we classify the epitopes recognized by the monoclonal antibodies that we develop in the lab as strain-specific, that meaning that those antibodies were recognized in epitopes which were present in certain strains but not in others, or they were group-specific, so some antibodies recognize all the strains from the same antigenic group, but not from A, but not from B, or vice versa, and also conserve epitopes, so that are conserving all the viral strains. And that's, and this is where we located the strain-specific epitopes in the C-terminal part, and that's why these epitopes were lost in those frame shift mutants that I described previously, because the sequence of this part of the molecule was changed in those mutants. Whereas the conserve and group-specific epitopes, they map in this central part of the ectodomain of the G protein, which is rather conserved. And were those changes that we found in the escape mutants also representative of changes that occur in vivo during the virus evolution? Well, at least some of them may be, okay? For example, some frame shift mutations were described by Solander and Gail Wirtz in this paper. They saw viruses that they had this type of frame shift mutations that I described. Also, it's common to find viruses that have premature or alternative stop codons. Okay. AG hypermutations are more difficult to identify, but I think we can also identify events that might be similar to the AG hypermutations that we saw in the escape mutants. And not only these drastic changes that we were seeing in the escape mutants, but even in natural isolates, we also identify other changes that were also very extensive, and they were occurring in viruses that were circulating. And we first identified these viruses that had a 60 nucleotide duplication. And these viruses, they have the name of BA, which means Buenos Aires. So they were isolated in this country, okay? And these viruses, they replicate quite well, not as well, so well that they spread all over the world, and now they have replaced all the previous viruses, and now they are the dominant viruses. Similar duplication events were also described for the group A viruses recently. So G protein is very weird. Can change almost essentially all and still be functional. And I mentioned that G protein is an extremely variable protein. Nothing like, it's the most variable protein that I know. And particularly in these variable regions in the ectodomain. And we published this was almost 25 years ago, that there was a correlation between genetic changes in the G protein and changes that we could detect with the monoclonal antibodies that we had available. So there was a correlation between antigenic and genetic changes. And we published this paper, and this paper has been referred by many people to as a demonstration, or at least an indication that there is a positive selection mediated by antibodies in the evolution of RSV. And we believe that until last year, that we published another paper in which we questioned that, okay? And obviously, I think it's still a big mystery why this extensive variation in sequence in the G protein, why and what for? It's an intriguing question that we still don't know what. But I think we can assess that we can say that if antibody selection is playing any role for RSV evolution, is not the type of antibody selection that one can see in other viruses, like, for example, in influenza virus, in the HA of influenza virus. It has to be some more subtle, and we don't understand. Well, so far for the G protein. Now we are going to go to the protein that Barney was referring to, which at the beginning we thought, it's a boring protein because it's behaving always the same, so as expected. So G protein was much more fun because we could predict anything and be sure that we were going to be wrong. So, but eventually, it turned out that F protein was important because all of the testing for all the antibodies that we have available in the lab for neutralization, those that neutralized best were those that reacted with the F protein. And since neutralizing antibodies was demonstrated by many people that they were correlated with protection against infection, we thought, well, we should also study the F protein, okay? So we switched to the F protein and tried to locate epitopes that were recognized by antibodies and so on. And in 1998, okay, we published this paper in which we more or less gathered together all the information that we had at that moment about the epitopes that we could map in the primary structure of the F protein, essentially. And we also speculated about secondary structure domains, okay? And that's what we came in that time. And this is more, this is an updated version, the last version of how the F protein looks like and where the antigenic sites are and so on. Well, you immediately realize that we were missing some resolution, a few angstroms resolution from here to there, all right? Well, I said that in our collection of monoclonals, those that neutralized best were those that recognized the F protein. But they were all murine monoclonals, and the question that we asked was, well, what is going on in the human situation? So which are the antigens that are recognizing the you are bringing some water? That's good. Which are the antigens that are being recognized by the neutralizing antibodies that are present in a human immunoglobulin preparation. Thank you. So what we did was to purify soluble forms of the F protein and the G protein. The F protein, we express it as the ectodomain of the molecule without the transmembrane region. And the G protein, there is a soluble form which is produced normally in infected cells. So we purify these forms and bound them covalently to Cephalus beads, made these columns. Then we took this preparation of immunoglobulins, RSV16, which for those that we are old enough, we know that this was the first medical product that was specific for RSV that was licensed. So it was an immunoglobulin preparation that was prepared from plasma of donors that were screened for high titers of neutralizing antibodies. And then we passed these immunoglobulins through this column or that column and collected the fraction that didn't bind to the columns or eluted the antibodies that bound, and tested for, checked that the depletion of the antibodies was efficient, and this we did by ELISA and it worked, and then we tested for neutralization, and this is a critical experiment. So we tested the neutralizing activity of this, the original material, or this material that went through this column, or this, or one that was went first through this column and then through the second column and didn't bind. Okay, and the result is shown here. Either the original preparation or these other depleted fractions, they neutralize essentially the same. So the answer to our question is, are these neutralizing antibodies recognizing the F, the G? And the answer apparently was no, none. Okay, so another mystery. So F, that was going to be an easy partner, well, it wasn't so easy after all, okay? We check also depletion by another assay. And so cells that were expressing the G protein, they were not recognized by the material that had been depleted with the G column, like, as indicated by these empty boxes, right? In contrast, the antibodies that went through the F column and they were depleted of antibodies binding to the protein that was used for preparing the column, they still recognized the surface of cells that were expressing the F protein. And this obviously, the conclusion was that there was something in the protein that was expressed at the cell surface which was different from the protein that was used for the column. And, but still we didn't understand what was going on, essentially because at that time we still were not so much aware of prefusion and postfusion protein. At that time, Bob Lamb published a paper which was very important. This was the first paper, or the paper in which they described the first structure of the fusion protein of any paramyxovirus. And they proposed this model for how the fusion of the viral membranes are mediated by the fusion protein of these viruses. So in the virus particle, the fusion protein, which is a trimer, is in a metastable conformation, which is called prefusion. And when the virus is going to infect the cell, this protein is activated by a mechanism that, in our case, we still don't know much how it happens. But anyway, once the protein is activated, it initiates a series of conformational changes, which include the formation of this prehairpin intermediate that I mentioned in one of the earliest slides. And refolding of this unstable intermediate brings into proximity the two membranes. At the end, they mix the lipids and form what is called a fusion pore, and the protein ends up in this structure, which is a very stable structure. It's called postfusion. So the protein goes from this prefusion metastable, high free energy conformation to this very stable post-fusion and low free energy level conformation. So there is a transit from a high level to low level free energy conformation, and that is the so this energy that is released is the one that is used to drive this process. So once the protein is activated, the process goes spontaneously because there is energy which is produced and is released on these conformational changes, which is used for the membrane fusion. So when we learn about this, we look at the protein that we had used for making the column that I referred before. And this is a three-dimensional reconstruction, EM reconstruction of the protein that we use for the preparation of the column that was used for the depletion studies. And this immediately, one could see that it resembles this structure and not that structure. So the conclusion was, well, maybe that the antibodies that we were depleting were directed against the post-fusion, and the neutralizing antibodies were mostly directed against the pre-fusion. So this was a very obvious hypothesis, but to prove that was not so easy. Again, it took us a number of years to get some form of pre-fusion F protein that we managed to stabilize by introducing certain mutations. I'm not going to go into detail, but eventually we finished with a protein that looked like by electron microscopy, albeit well, different, completely different than the post-fusion that we had already familiar with. And this protein resembled the structure that it was described by Paul Blanc. Eventually, we proved that molecule, which is this one, is able to bind the antibodies that were not depleted in the column and that maintain most of the neutralizing activity. Okay. So this was a very good surprise, and the question was, well, so how this pre-fusion protein looks like, and what are the antigenic sites in that molecule? For that, our friends Jason and Barney and Peter Kwon, they came and they managed to express the protein, to engineer, to stabilize the molecule in the pre-fusion conformation, to get crystals and solve the structure. So they save us a lot of work. This is the structure that they got. This is the trimer in the pre-fusion conformation, and they had already solved the structure in the post-fusion. And here is the one of the protomers in the pre-fusion and the post-fusion, and you can immediately realize the dramatic changes that happen when the protein transits from this conformation to this other conformation during the process of membrane fusion. And more important, we could use now this structure to locate the epitopes recognized by antibodies that were that had certain properties, for example, neutralization. And they were also able to describe new antigenic sites that had not been previously recognized, that were specific for the pre-fusion conformation, that they they recognize a site which is not in the post-fusion conformation, that's called site 0, and these antibodies turn out to be the most potent neutralizing antibodies described so far. That explains why the antibodies that we were depleting of the post-fusion with this form of the protein still retain most of the neutralizing activity. Okay? Besides the antibodies being relevant for vaccines, for example, and of course we are interested on that, we are also interested in biology, in the biology of the virus. And for that, the antibodies, monoclonals, are unique reagents. They are fantastic reagents because they have a very exquisite specificity. And I'm going to give you a couple of examples. Again, oops, sorry. Jason, with Barney and Morgan, that she's sitting somewhere there, okay, they managed to get this complex of the pre-fusion F protein bound to this antibody, AM14. And this antibody is very special because it recognizes or interacts with residues, amino acid residues, from two adjacent protomers, which are in the pre-fusion conformation of the molecule, not in the post-fusion. So what we found, we tested with some preparations that we had in the lab with the pre-fusion and the post-fusion. This antibody binds to the pre-fusion quite well, but only after cleavage, and you will hear about that in a minute. Okay, so when the protein is cleaved, it binds very well AM14. When the protein is not cleaved, it doesn't bind, even if it is in the pre-fusion conformation. Pre-fusion, as long as we say, for example, how the two proteins bind to this other antibody, D25, which is one of the antibodies that bind to site 0, and so it's specific for the pre-fusion and not or reacts very poorly with post-fusion in blue here. Okay, so this antibody can it identifies a structural change that happens when the protein is cleaved, and this is important because we described also years ago that all these fusion proteins from paramyxoviruses, viruses, but also from other viruses, they require cleavage for being active in membrane fusion. And in most cases, the cleavage occurs only once, just before this hydrophobic region, which is called the fusion peptide, which is the part of the molecule which is inserted in the target membrane. RSV is unique in this respect, in the sense that it has two cleavage sites. And why two cleavage sites? I have no idea. What's the relevance of these cleavage sites from the structural and functional point of view? We still don't know. But the antibody that I just mentioned, AM14, is going to be a very useful reagent, at least to explore some possibilities. The other example is this one with an antibody that we isolated recently in the lab. It's called 64PF. Remember that I told you we always say F or G. Now it's F, but it has a P because it's pre-fusion. Okay, so look at how this antibody neutralizes virus. This is the brown line is the palivizumab, Synagis. Okay, so this is better, not as good as D25, which is the one that is specific for site 0, but it's quite good. D25, of course, binds, I already said that it binds very well to the pre-fusion conformation, poorly, very poorly, or not at all to the post-fusion. But this antibody doesn't bind pre-fusion, post-fusion, doesn't bind anything, neither this form or this form. And yet this antibody binds to the cells that are, bind to the surface of cells expressing the F protein. And it binds to those cells, and we could isolate escape mutants that are resistant to the neutralization by this antibody, and these mutants, now this mutant here, that's the line, you see is neutralized by other antibodies, but not by this antibody, and this mutant has mutation here. So here is another structure, which apparently is different from this one and this one, probably is very similar to this one, but it's slightly different, and that's in the surface of cells. So there's another structure for Jason to solve, okay? Finally, just a few words about the cousin of RSV, which is the metapneumovirus. Also, in collaboration with Jason, we managed to express and stabilize a stabilized form of the post-fusion conformation of metapneumovirus. And the structure of one of the protomers in the trimer is shown here in white, and it is in comparison with the structure of the similar structure of the F protein of RSV in blue. You can see that they are almost identical. And even the sites that are relevant for antibodies that neutralize, they are also very, very similar. There are more differences when they are this structure is compared with the equivalent post-fusion of the other paramyxoviruses. of viruses. Given this similarity in the structures, we decided to check whether any of the antibodies that we have in the lab were able to cross-react and cross-neutralize the two viruses. And in fact, we identified this antibody, 101F, that we had isolated years ago in the lab. And this antibody binds, as you can see here in the EM pictures, it binds to the protein, the post-fusion form of the RSV and the post-fusion form of the metapneumovirus fusion protein. So this antibody cross-neutralize and cross-react and cross-neutralize the two viruses. And yet, despite this cross-neutralization with this monoclonal, and some other reports about other sporadic monoclonals that bind and cross-neutralize the two viruses, still in a polyclonal response, if you immunize with the protein of metapneumovirus, you get good binding to the metapneumovirus protein, but not to the RSV, and vice versa, and also in the neutralization assay. So in these cross-neutralizing antibodies, they probably represent a very small minority in the population of the antibodies that are raised in a polyclonal response. So to finish, I would like to acknowledge all the people that have contributed to my career. And I try to put all together in this picture, okay? Even some people are left out, okay? But to single out at least those that had collaborated more recently, of course, Jason has been an excellent, we collaborate not only in science, but also in football. We had our discussions, Skype discussions about football. Barney, who really pushed us to start the collaboration. Ron and Bernadette had been also very helpful to start in the lab to work with metapneumovirus. Geraldine has been a long-standing collaborator for many years. These two people are electron microscopists that have helped us a lot in recent studies. And Rosa Rodriguez is a pediatrician in Madrid. We started collaboration recently. And also in the lab, there are many people that have been in the lab. So I brought this picture from a few years ago. We look like a football team, you know? And a more recent one in which you can see some faces are still there and others have changed. And last but not least, these are people that had also a very high impact factor in my career, okay? So finally, Peter Collins and I wrote a review article some years ago, and well, to be fair, he wrote the article, okay? I put my name into it, okay? And the second part of the title, he wanted to introduce it because Paul Simon, right? Still crazy after all these years. So that's more or less how I feel today. Thank you very much.